Assalamu alaikum. Today we will talk about the mechanisms by which the killing of microorganism takes place in the phagosome. In the previous video we talked about inflammation and the phagocytosis. Soon after the engulfment of the microbe, a new metabolic pathway happens. This pathway is known as respiratory burst. It triggers the uptake of oxygen by the cells. So, due to the respiratory burst, two important microbicidial agents are produced. First one is the superoxide radical, and second is hydrogen peroxide. These highly reactive compounds are synthesized by the following reactions. So you can visualize these reactions. In the first reaction, molecular oxygen is produced by an electron to form the superoxide radical. This superoxide radical is a weak bactericidal agent. In the next step, the enzyme superoxide dismutase catalyzes the formation of hydrogen peroxide from two superoxide radicals. Hydrogen peroxide is more toxic than the superoxide but is not effective against catalase producing organisms. Some of the microorganisms such as staphylococci produce an enzyme which is known as catalase. Catalase enzyme is a protective enzyme that degrades hydrogen peroxide into water and oxygen. So the killing of organisms within the phagosome is a two-step process that consists of degranulation followed by the production of hypochlorite ion. In the previous video we talked about the engulfment of microbe. The microbe was engulfed by the neutrophils and macrophages in a vacuolar form, the form of a vacuole. That vacuole which contained the microbe was called as phagosome. Now the lysosomes that are present in the neutrophils and macrophages, they will fuse with the phagosome and they will release their content into that phagosome, and their content includes some of the enzymes, the degradative enzymes that will kill the microbe. So the granules that are present in the neutrophil are known as lysosomes, and there are two types of granules. There are some large granules which are called as large lysosomal granules, and some are small granules. The large lysosomal granules, which constitute about 15% of the total, contain the important enzyme myeloperoxidase, as well as lysosomes and several other degradative enzymes. Myeloperoxidase, which is green in color, and it makes a major contribution to the color of the pus. Then there are smaller granules which make up about remaining 85% of the granules. They contain lactoferrin and additional degradative enzymes such as proteases, nucleases, and lipases. Lactoferrin are enzymes that chelate iron from the bacterial cells. So lysosomal granules can empty into the extracellular spaces as well as into the phagosome. Outside the cell, the extracellular enzymes that are secreted by the macrophages outside the cell, those enzymes provide uh, defense against some fungal cells, fungal hyphae, and also against some of the parasites such as roundworms and tapeworms. Because those structures are so much larger than the neutrophils and the macrophages that the cells cannot engulf them, so extracellular enzymes are produced which are excreted outside the cell to kill those pathogens. The actual killing of microorganisms occur by a variety of mechanisms which fall into two categories. One is the oxygen dependent reactions and second is the oxygen independent reactions. First we will talk about the oxygen dependent reactions, then we will go through the oxygen independent reactions. The most important oxygen dependent mechanism is the production of highly reactive hypochlorite ion by myeloperoxidase. Following reaction describes the production of hypochlorite ion. In this reaction, chloride ion plus hydrogen peroxide. The hydrogen peroxide was produced by the respiratory burst. This yields hypochlorite ion in the presence of myeloperoxidase. Chloride ion plus hydrogen peroxide produces hypochlorite ion plus water. The hypochlorite ion by itself damages cell walls but can also react with hydrogen peroxide to produce singlet oxygen. In the first reaction, the chloride ion reacted with hydrogen peroxide to produce hypochlorite ion. Then the hypochlorite ion again reacted with the hydrogen peroxide to produce singlet oxygen. And this singlet oxygen damages cells by reacting with double bonds in the fatty acids of the membrane lipids. Now let's move towards the oxygen independent mechanisms. Oxygen independent mechanisms are important under anaerobic conditions. These mechanisms involve lactoferrin, which chelates iron from the bacteria, lysosomes, which degrade peptidoglycan in the bacterial cell wall, cationic proteins, which damage bacterial membranes, and low pH. So this was the discussion about the oxygen dependent and oxygen independent reaction. We have discussed about those mechanisms that were taking place in neutrophils, especially we were talking about neutrophils. Now we will look about the macrophages. Macrophages also migrate, engulf and kill bacteria by using essentially the same processes as the neutrophils do, but there are several differences. First difference is that macrophages do not possess myeloperoxidase and so cannot make hypochlorite ion. They do produce hydrogen peroxide and superoxide by respiratory burst. Respiratory burst is the increased uptake of oxygen by the cells. Second difference is that certain organisms such as the agents of tuberculosis, brucellosis and toxoplasmosis are preferentially ingested by macrophages rather than by the neutrophils, and they may remain viable and multiply within these cells and produce granulomas. Granulomas formed during these infections contain many of these macrophages. The third difference is that macrophages secrete plasminogen activator, an enzyme that converts the proenzyme plasminogen to the active enzyme plasmin, which dissolves the fibrin clot. Several bacteria can produce clots which help them disguise and that allow them to escape from the phagocytic actions of the cells. This was all for today. Thanks for watching. Allah Hafiz.